Disaccharides are carbohydrates that yield 2 monosaccharide units on hydrolysis. The hydrolysis of a disaccharide may yield 2 molecules of the same or different monosaccharide. In disaccharides, the 2 monosaccharide units combine and result in the formation of an oxide linkage with the elimination of a water molecule. For example, sucrose with 2 monosaccharides, glucose and fructose, joined together by an oxide linkage is shown in the structure here. Such a linkage, formed when the hydroxyl group of the hemiacetyl carbon of one monosaccharide condenses with the hydroxyl group of another monosaccharide, is called glycosidic linkage. In other words, a linkage between 2 monosaccharide units through an oxygen atom is called glycosidic linkage. The 3 most important disaccharides are sucrose, maltose, and lactose. Let us now focus our attention on their structural aspects. The well-known disaccharide plus sucrose or table sugar is obtained from sugarcane or sugar beets. It has the molecular formula C12H22O11. The hydrolysis of sucrose yields an equimolecular mixture of D plus glucose and D minus fructose. Solutions of sucrose are dextrorotatory, but when sucrose is hydrolyzed, it becomes levorotatory. There is an explanation for this anomaly. The optical rotation of D-glucose is found to be plus 52.7 degrees, and that of D-fructose is minus 92.4 degrees. That is, D-glucose is dextrorotatory with an optical rotation of 52.7 degrees, while D-fructose is levorotatory with an optical rotation of 92.4 degrees. As the magnitude of levorrotation of fructose is greater than the magnitude of dextrorotation of glucose, the mixture obtained by the hydrolysis of sucrose is levorotatory. The sign of optical rotation changes from dextrorotatory to levorotatory during the process of hydrolysis. This is called the inversion of sugar. The levorotatory mixture, which is a mixture of equimolar glucose and fructose obtained from the hydrolysis of dextrorotatory sucrose, is called invert sugar. Invert sugar is the main ingredient of honey. Let's look at the structure of sucrose in greater detail. Sucrose is made up of 1 unit of glucose, specifically the alpha-anomer alpha-D-glucopyranose, and 1 unit of fructose, specifically the beta-anomer beta-D-fructofuranose. These 2 hexose units are joined by a glycosidic linkage that is formed between the C1 of alpha-D-glucopyranose and the C2 of beta-D-fructofuranose. Note that the loss of a water molecule results in the formation of 1,2 glycosidic linkage or alpha-beta glycosidic linkage. Thus, the 2 carbonyl functional groups of glucose and fructose are blocked with the formation of glycosidic linkage. As the reducing group is necessary to give Tollens and Felling's tests, is involved in the formation of the glycosidic linkage, sucrose gives a negative test with these reagents. Hence, sucrose is called a non-reducing sugar. Maltose is another important disaccharide which contains 2 glucose units. Its molecular formula is C12H22O11. It is the least common naturally occurring disaccharide. It is found in germinating grain. The glycosidic linkage in maltose is formed between the carbonyl group of the C1 of alpha-D-glucose and the hydroxyl group of the C4 of the second alpha-D-glucose. The availability of a free aldehydic group at the C1 of the second glucose in its solution allows the maltose to reduce Tollens and Felling's reagents. Hence, maltose, unlike sucrose, is a reducing sugar. Lactose is a disaccharide present in milk, so it is sometimes referred to as milk sugar. Lactose is composed of beta-D-galactose and beta-D-glucose. The glycosidic linkage in lactose forms between the C1 of galactose and the C4 of glucose. Like maltose, lactose is a reducing sugar due to the presence of a free aldehydic group at the C1 of the glucose unit. Can you answer this question on your own? What are the products expected when lactose is hydrolyzed? Lactose would produce galactose and glucose in equal proportions. Here, we see the Haworth projections of lactose and the hydrolysis products. Polysaccharides contain many monosaccharide units joined together by glycosidic linkages. Polysaccharides are the most common naturally occurring carbohydrates. A polysaccharide has the general formula C6H10O5 taken n times. They play important roles in food storage and as structural material. Cellulose and starch are the most important polysaccharides. Starch is the main storage polysaccharide of plants. Starch is found in cereals, roots, tubers, and some vegetables, and is used as an important dietary source for human beings. Chemically, starch is a polymer of the alpha isomer of D plus glucose. It consists of two components: amylose, which makes up 20% of starch, and amylopectin, which makes up the remaining 80%. These two components differ in their molecular size and shape. Amylose is the water-soluble portion of starch. Amylose molecules contain 200 to 1000 alpha-D plus glucose units, connected by C1, C4 glycosidic bonds, as shown in the illustration. The chains of amylose are long and unbranched. Amylopectin, the major component of starch, is insoluble in water. Amylopectin is a branched chain polymer of D-glucose units. The branched structure consists of several hundred short chains of about 20 to 25 D-glucose units each. An amylopectin molecule may contain as many as 1 million glucose units. The main chain of amylopectin contains alpha-1, 4 linkages between the glucose units, while branching occurs by alpha-1, 6 linkages, that is, C1 to C6 glycosidic linkages. Consider the illustrations here. Which one best represents amylose? Which one best represents amylopectin? You just learned that amylose is an unbranched chain, whereas amylopectin is branched. Therefore, picture A shows amylopectin with its branched chains. Picture B shows amylose. Note that the unbranched chains of amylose often adopt helical structures. Cellulose is another important polysaccharide. Found exclusively in plants, it is the most abundant of all carbohydrates. Cellulose is the chief structural material in plants. It is completely insoluble in water. Cellulose is a straight-chain polysaccharide. It is made up of chains of beta-D-glucose units. These beta-D-glucose units are joined by glycosidic linkages between the C1 of one glucose unit and the C4 of the next. These are also called beta-1,4 linkages. Humans cannot digest cellulose, as they don't have the enzymes necessary to do so. Cows, horses, goats have symbiotic bacteria in their intestinal tracts. These bacteria provide the enzymes for breaking down cellulose. The term dietary fiber refers to cellulose. The polysaccharide, glycogen, is the form in which carbohydrates are stored in animals. It is present in the liver, muscles, and brain. Glycogen is also found in yeast and fungi. The structural aspects of glycogen are much similar to amylopectin, a component of starch. Hence, it is referred to as animal starch. The structure of glycogen differs from that of amylopectin in that the molecules of glycogen have a greater degree of branching. Enzymes break down glycogen that is stored in the liver and muscles into glucose when the body needs it. Now let us study briefly the importance of carbohydrates in our lives. Carbohydrates are a very important class of biomolecules. They form a major component of our diet. They function as storage molecules, starch in plants, and glycogen in animals. The cell walls of bacteria and plants are made of cellulose. Carbohydrates have many uses as structural material. We build houses and furniture and other useful items from wood, which is 40 to 50% cellulose. We clothe ourselves in cotton and linen, which are largely cellulose. Carbohydrates provide the basis for a variety of industries, including textiles, paper, lacquers, and breweries. Carbohydrates have a number of important biological roles. The sugars D-ribose and 2-deoxy-D-ribose are components of nucleic acids. Glycoproteins and glycolipids, that is, proteins and lipids with attached carbohydrate groups, carry out a variety of important biochemical functions. The word protein is derived from the Greek word proteos, which means primary or of prime importance. Proteins have a number of important biological roles, both structural and functional. They are vital for the growth and maintenance of the body. The dietary sources of protein include milk, cheese, pulses such as beans and lentils, peanuts, fish, and meat. Proteins are polymers of alpha amino acids. Now, what are amino acids? Let us understand. Amino acids are compounds that have one or more amino groups and carboxylic groups in a molecule. On the basis of the relative position of the amino group with respect to the carboxyl group, amino acids are classified into alpha, beta, gamma, and so on. Examples of each class are shown here. In alpha amino acids, the amino group is present on the alpha carbon atom. Similarly, in beta and gamma amino acids, the amino group is present on the beta and gamma carbon atom respectively. An alpha amino acid is illustrated here. R represents the side chain. Alpha amino acids are the building blocks of proteins. Only alpha amino acids are found in naturally occurring proteins. There are 20 odd alpha amino acids, as shown in the table. In nature, 20 odd different amino acids combine in different ways to form different protein molecules. Every alpha amino acid has a common name. These common names reflect either the source or the property of that compound. Alpha amino acids are usually represented by a 3-letter symbol and sometimes by a 1-letter symbol. For example, the amino acid glycine has a sweet taste. Its name is derived from the Greek word glykos, which means sweet. The 3-letter code for glycine is GLY, and its 1-letter symbol is G. The side chain of glycine is a hydrogen atom. The amino acid triosine was first obtained from cheese. Its name is derived from the Greek word tyros, which means cheese. The side chain for tyrosine contains an aromatic phenol group. It has the 3-letter symbol TYR, and its 1-letter symbol is Y. Among the natural amino acids, the ones that can be synthesized in the body are called non-essential amino acids, whereas those that cannot be synthesized in the body and therefore must be included in the diet are called essential amino acids. Some examples of non-essential amino acids are alanine, serine, and tyrosine, while examples of essential amino acids are histidine, tryptophan, and methionine. Amino acids can be classified as acidic, basic, or neutral on the basis of their nature. An amino acid is acidic if it contains more carboxylic groups than amino groups. It is a basic amino acid if it contains more amino groups than carboxylic groups. Neutral amino acids contain an equal number of amino and carboxylic groups. Can you classify these amino acids as acidic, basic, or neutral? Look carefully at the structures. Molecules A and E, which are aspartic acid and glutamic acid, are acidic. Each has 2 carboxylic groups and 1 amino group. Molecules B and C, which are arginine and lysine, are basic. They have multiple amino groups but only 1 carboxylic group. Molecule D, the amino acid valine, is neutral. It has 1 amino group and 1 carboxylic group. Let us now study the physical properties of amino acids. In general, amino acids in their pure states are colorless, crystalline solids. They are water-soluble with high melting points. Most amino acids behave like salts rather than like simple amines or simple carboxylic acids. Their behavior, like salts, is attributed to the presence of both the acidic carboxylic group and basic amino group in the same molecule. In aqueous solutions, the acidic carboxylic group of an amino acid transfers a proton or a hydrogen ion to the basic amino group present in the same molecule. This results in the formation of a dipolar ion called a Zwitter ion. Thus, amino acids form Zwitter ions in aqueous solutions. Zwitter ions are electrically neutral. They exhibit amphoteric behavior as they can react with both acids and bases. The high melting points and solubility of most amino acids are due to this dipolar ion structure. All alpha or naturally occurring amino acids, with the exception of glycine, are optically active due to the presence of an asymmetric alpha carbon. Glycine does not have an asymmetric carbon and is therefore not optically active. All amino acids except glycine are chemically correlated to level rotatory glyceraldehyde with L configuration. Hence, amino acids have the configuration L. Notice that in L configurations, the amino group is drawn on the left. Proteins are a class of biologically important compounds. They are condensation polymers of about 20 different alpha amino acids, which are linked by peptide bonds. A peptide bond is formed when the amino group of one amino acid molecule interacts with the carboxyl group of another amino acid molecule. This reaction involves the elimination of a water molecule. Thus, the interaction between the amino group and the carboxyl group of two of the same or of different amino acids results in the formation of an amide linkage, that is, an NHC double bond O group called a peptide linkage. The product formed when two amino acids interact is called a dipeptide. The name dipeptide indicates a peptide bond that is formed between two amino acid molecules. For example, when glycine combines with alanine, a dipeptide, glycylalanine, is formed. Similarly, when two glycine molecules combine, glycylglycine is formed. Note that, by convention, the amino end is shown on the left and the carboxyl group is on the right of the resulting dipeptide. Also note that the peptide bond is planar. The CON and H atoms are all in the same plane along with the alpha carbons. Let's work on a practice problem. Show the formation of the peptide bond that forms between alanine and serine. Keep the amino acids in the order given here. It may help to start with the peptide backbone. Then look up the side chains and include them in the correct order. The alanine serine, represented as aliser dipeptide, results. Three amino acids linked by two peptide linkages are called tripeptides. The tripeptide glutathione protects the cells from oxidizing agents. It is formed from glutamic acid, cysteine, and glycine. Similarly, when 4, 5, or 6 amino acids are linked, the products obtained are called tetrapeptide, pentapeptide, and hexapeptide respectively. When more than 10 amino acids are linked, the product obtained is called a polypeptide. Thus, depending upon the number of amino acid residues present per molecule, peptides are classified into dipeptide, tripeptide, tetrapeptide, and so on up to polypeptides. Polypeptide chains that have either more than 100 amino acid residues with a molecular mass greater than 10,000 or less than 100 amino acid residues with a well-defined conformation are referred to as proteins. Let us now look at the classification of proteins. On the basis of molecular shape, proteins are broadly classified into two classes: fibrous proteins and globular proteins. In fibrous proteins, the polypeptide chains are long and thread-like and run parallel to each other to form fibers. These parallel chains are held together by hydrogen bonds and disulfide linkages. Due to their strong intermolecular forces of attraction, fibrous proteins are generally insoluble in water. Important examples of fibrous proteins include keratin, found in hair, nails, skin, etc. Myosin, found in muscles. Collagen, found in tendons. And fibroin, found in silk. Thus, these proteins form the structural material of animal tissues. In globular proteins, the polypeptide chains coil around to give a roughly spherical shape. These are soluble in water due to the presence of hydrophilic groups. Globular proteins maintain and regulate life processes. For example, insulin regulates the blood sugar levels in the body. Hemoglobin and albumin are some other examples. Let us now discuss the three-dimensional structure of proteins. The structure of proteins is studied at 4 different levels, in the ascending order of their complexity, as primary, secondary, tertiary, and the quaternary structure. Let us first discuss the primary structure of proteins. The specific sequence of amino acids in the polypeptide chain reflects the primary structure of proteins. Here, we see the specific arrangement of amino acids of the enzyme lysozyme. Every protein has its own unique amino acid sequence. Any change in the sequence of amino acids creates an altogether different protein. The secondary structure of proteins refers to the arrangement of the polypeptide chains in space. One important type of secondary structure is the alpha helix, illustrated here in a ribbon diagram. Each polypeptide chain is coiled to form a spiral structure called alpha helix. The alpha helix is a right-handed helical chain in which the hydrogen bonding occurs between different pairs of the chain to hold the helix together. The hydrogen bonds occur between the oxygen atom of the carbonyl group of one amino acid residue and the hydrogen atom of the NH group of a different amino acid residue on the adjacent turn of the helix, as shown here. Another common secondary structure in proteins is referred to as beta-pleated sheet structure. The beta-pleated sheet is nowadays referred to as the beta structure. The arrows in the ribbon structure here show the beta structure. The contraction of the peptide chains results in a pleated sheet. The chains lie side by side and are held together by hydrogen bonds. Such a structure is called a beta structure or arrangement. The tertiary structure of proteins describes the further folding of the secondary structure. The tertiary arrangement of helices and sheets is held together by hydrogen bonding, disulfide linkages, van der Waals forces and electrostatic forces of attraction. Shown here is the tertiary structure of proteins. The further folding of the secondary structure gives rise to two major molecular shapes, fibrous and globular proteins. Let us now look at the quaternary structure. Some proteins consist of multiple polypeptide chains called subunits. The quaternary structure describes how the subunits are arranged in space relative to one another in the complete protein. Hemoglobin is an important example. As shown in the illustration, the 4 protein chains of hemoglobin are arranged in a compact bundle. A protein found in a biological system with a unique structure and biological activity is referred to as a native protein. In some respects, proteins are rather fragile. Temperature or pH changes can cause a disruption of the hydrogen bonds. When hydrogen bonds break, the globules unfold, the helices unravel, and the beta sheets flatten out. This is called protein denaturation. When proteins denature, the primary structure, the specific amino acid sequence, remains intact. However, the secondary and tertiary structures come apart. Denatured proteins are unable to carry out their biological function. If you have ever boiled an egg, you might have witnessed protein denaturation as the egg white solidifies. Enzymes are proteins that catalyze reactions, speeding up the reaction rate. Enzymes speed up reaction rates by reducing the activation energy. Most enzymes are globular proteins. Enzymes catalyze specific reactions regulating a variety of functions in the body. Enzymes are exquisitely sensitive, acting only on a certain substrate or class of substrates. Most enzyme names contain the suffix ase. Enzymes may be named after the substrate they act on. For example, the enzyme maltase catalyzes the hydrolysis of maltose to form 2 molecules of glucose. Alternatively, enzymes may be named after the reaction they carry out. Dehydrogenases or oxidases carry out redox reactions. Isomerases catalyze isomerization reactions. Hydrolases make hydrolysis reactions faster.